Thanks. Thanks everyone. Great to be here. So this is going to be mostly a sort of theory-based talk on some work, a mathematical limit I worked on with my PhD advisor, Jeff Vassell, back in Edinburgh. And I'm going to start by sort of motivating it in the context of biological systems, biological networks. And I'll finish with some preliminary work I've started with Eleni Kadafori's group at the Flatiron Institute. And the idea is to kind of motivate discussions about where maybe these kind of ideas could apply to your own biological systems networks. Okay, so let's get started. So biological networks, everyone's got their own sort of canonical example in mind. Here we've got the classic case of leaf venation, this classic sort of hierarchical patterning. We've got the fly brain, fly connectome over here, relatively recent, incredible segmentation. We've got someone's won their local lily pad growing competition. Yes, this is a real thing. We've got the whole mouse brain vasculature. There's a relatively new projected light sheet microscopy technique out of Columbia University, which apparently has really increased the efficiency for these kinds of networks. And then lastly, we've got a fluorescent imaging of a chick embryo by a collaborator, Liz Jones at KU Leuven, who we're now working on with actually quail data. So to understand these networks, often understanding the local phenomena is obviously crucial. So in this case, we're looking at blood flow networks. We've got David Barlow here, who's a hobbyist who does these incredible imaging of various biological systems. And here, if we're going to try and understand things like clotting, we clearly want to get the local details as accurate as possible. We want to take into account the heterogeneity of the material and as much as possible. And people have done this. So Peter Barlow at NJIT has done amazing simulations in his group, taking into account as much of the details as possible. These are incredible feats, but extremely computationally expensive and are very hard to scale up. And as we know, I really like this quote from Biot et al. So from the mechanical point of view, microcirculation is an extraordinarily highly admirable system. So imagine you have 4 to 5 liters of blood, a concentrated suspension of 3 types of cells of varying sizes, shapes, and stiffness, also carrying proteins and molecules circulating in a closed, interconnected, hierarchical network feeding each cell of each organ. A single pump is producing the forcing, and its length is some 80,000 kilometers in the human. It turns out quite recently, Poole et al. Updated this figure from, it was August Kroc in 1919, made the first sort of estimation of the total length of the microcirculation, and they worked out that one of his assumptions about capillary density in various areas was quite a way off. So it turns out, it's more roughly 16,000 kilometers in total length. So how do people get around this in the context of these vascular networks? So Quattroni has a really nice review article about essentially trying to reduce complexity where appropriate. So clearly we have a massive range of scales in the vasculature. So for things like the heart and the major arteries, major veins, we need quite a comprehensive model. So people have these fluid structure interaction problems, which are quite computationally expensive, in line with sort of what we saw before. As the complexity reduces down into the sort of arterioles, capillaries, venules, you can start making assumptions about the flow, linearizing Navier-Stokes and assuming angular symmetry, things like this, and you can reduce it to 1D equations along the vessels. And then finally, there are certain areas where you can sort of treat it as a black box, and we get rid of all spatial dependence. But even here, he talks about this virtual physiological human project based in Belgium, I forget which institute, which is trying to model just about every aspect of the human possible. They go through the details of how essentially, as making all the assumptions possible here that they can think of, it's still way out of touch with sort of modern computational capabilities. And so where I sort of come in is I'm wanting to think about large-scale dynamics at this sort of like one-dimensional scale in biological networks. So with all the complexity and detail in these systems, why should we expect large-scale eigenmodes to be relevant? Well, the idea is that the small-scale details are clearly always going to be important, but large-scale linear dynamics persist after the system has time to settle down. So, classic example, particularly relevant back home in Australia, everyone's always talking about whether we're in a El Nino, La Nina cycle. So the weather, obviously an extremely complicated system, it still has these sort of persistent, coherent, large-scale dynamics. And here we have on the order of sort of every half a decade, 5 years, this, large-scale oscillation in weather patterns between Southeast Asia, South America. And then a more sort of like, modern example we've got with these incredible new data sets increasingly coming out that various people in this room have done amazing work. We're starting to find that the connectomes, when we look at the eigenmodes, they're informing large-scale functionality. And I'm investigating this kind of area. Okay, so the system I look at, that we've been looking at, is we start with a network. So here I've got a triangulation of the sphere, and on each edge, parametrized by the one-dimensional coordinate x, edge E, we look at this classic eigenvalue equation for the Laplace operator. And so this will give you things like the spatial dynamics of the like a vibrating string, diffusion, and the sort of reduced Schrodinger equation. It's actually in this context that this system really sort of started being investigated by Linus Pauling in, I think it was the 20s, 30s. And so the vertices, we have continuity. So eigenfunctions f on edges adjacent to each other at a particular, incident to each other at a particular vertex need to match, and then we've got Kirchhoff's law. So the sum of the first derivatives is 0. Here I'm going to plot a superposition of eigenmodes over the whole network for the wave equation. And so here, color and sort of distance from the origin are representing the same thing, a bit laggy, but this is just mode amplitude. So the idea is we've clearly got this large scale dynamics on the scale of sort of the entire network, but you can roughly pick it up. We've also got these small scale dynamics on the scale of an edge. And what we've investigated is there some continuum description of what's going on over the entire network? And especially in the limit, is there some PDE, for instance, that reduces all these edgewise equations to a single equation on the underlying embedding space, which is in this case the sphere. Okay, so just to briefly go through how we solve this in general. So locally, we have some edge length L v w, connecting vertices v and w. We solve this as a standard undergraduate exercise, find the trigonometric solutions. And once we feed this into the vertex conditions, forcing things to match with continuity and Kirchhoff like before, we get this condition, which is a sum over neighbors W of the vertex V, and we have the spectral parameter K. And because of continuity, we can just say that once you get to a vertex, there's just a single value. You set this up as a system over the entire vertex set, capital V, and you can solve this numerically. You get this sort of infinite increasing sequence of eigenpairs, and that is what I solved to produce this animation before and plotted just some superposition. And the question then is, what happens at the continuum limit? The details of all this are in a paper in review at the moment, which you can check out or obviously ask me about. But essentially, you get this PDE in the limit to order length squared. And this now, the x coordinate is living on the embedding space. And it essentially has two critical components that we introduce. So the first thing is this edge tensor. And this essentially is a sum. It's evaluated at locally at vertices. And it's a sum over edge vectors tensored with themselves. So it might look slightly like the pneumatic order tensor if you know anything, if you have experience with liquid crystal theory. And so this is one of the components. Its trace is just then the sum over edge lengths. And for example, in this particular case, on this lattice, square lattice, it just becomes a multiple of the identity. So for a homogeneous and isotropic graph, we just get this multiple of the identity. And then we also have this vertex number density mu, which is sort of the empirical number density of nodes. And in practice, you can sort of think of it like a inverse cell area from finite elements. So that's what it comes out as in this particular case. And I don't know if that's coming up very well, but here are a bunch of examples where we're comparing the graph solutions, the numerical solutions that I calculated before, showed how to calculate before with this matrix problem, and then at the bottom are the PDE solutions. So here we have this sort of spiderweb looking network. The continuum limit just comes out as some sort of arbitrary PDE once you fill in, substitute in all the values for mu and r. In the case of a homogeneous and isotropic network, so here we've got square, hexagonal, and then cubic, the limit, once you substitute in for this multiple of the identity before, the limit actually just becomes the standard sort of d-dimensional Laplace operator. But you get this interesting factor of the embedding dimension that comes out. And then here we've got a rectangular lattice, which produces a new PDE. Now, clearly these aren't particularly biologically relevant networks. In practice, things are very noisy, and we find that even for relatively innocuous-looking networks, we can get limiting tensors which don't have a well-defined limit as we decrease the edge lengths. And especially here we can actually explicitly calculate the tensor, but it's very unclear how to proceed when we have something more biologically relevant. And so we've, don't worry too much about the details here, but there's this homogenization theory which people use to study, say, like diffusion or flow in porous media. And it essentially consists of separating scales. So we can separate scales before by essentially considering sort of homogenized patches of the graph, solving a particular sort of patch problem, which we construct here, and then coming up with like an effective description of what's going on in the network. And we've applied this to a range of networks. We've got this sort of random neighborhood graph, the triangulation from originally, and then we've got this aperiodic monotile here. And we find that we still get out, as long as the graph is statistically homogeneous, we get out this Laplace operator rescaled, but we get a sort of effective dimension. Before we were finding 2 or 3 in the case of the cubic lattice, and here we have some effective dimension. And we can generalize this then to more interesting networks with heterogeneity and density. And we now have a sort of well-formed theory for how to try and take continuum limits of these dynamics on more biologically relevant structures. Okay, so lastly, in the last few minutes, I'll go through sort of a summary of some work that I've been doing recently at the Flatiron Institute. So here we've got, Liz has done this fluorescent imaging of the quail embryo and extra-embryonic vasculature here. And this is sort of an illustration of what it actually looks like in practice. And so basically what we want to study is the flow and the structure of the network. So we've got this amazing imaging here. We've got the heart beating here. We've got this arterial flow here and this sort of peripheral venous flow here. And so a first approach we've made is to derive a damped wave equation for the pressure, which involves a whole lot of simplifying assumptions, which we think apply at the larger scales. And the crucial parameter here is this function of the radius of each vessel, and it has things like the Young's modulus and vessel thickness. Vessel wall thickness, we've got continuity, flow conservation at the nodes, and then pulsatile forcing at the heart. And so here is a drawing by Frank Lilly, 1936, of the different components of the vasculature at this stage. And here, I've got some preliminary results. This is a sort of non-dimensionalized pressure magnitude starting from the heartbeat here and then pulsing through the entire network. And so what we want to try and match this to is the data that I was showing before and try and characterize these large-scale coherent waves and look at things like the impact of the structure of the network and then more sort of, different dynamics such as sort of the arterial venous differentiation. And yeah, that's it. So thanks a lot.
Any questions for that interesting talk?
Thank you so much for the very interesting talk. Yeah.
Hi.
And I'm sorry if it's my misunderstanding. I'm wondering if you've thought about maybe nearest neighbors kind of expansions to the formalism, maybe in like higher dimensions or something like that?
So it's maybe like a renormalization. So you start at a neighborhood and you're sort of clumping nodes together in like a subgraph and then iterating on that, or you're just saying instead of edges, yeah, okay.
Like long distance connections, right?
Long
like
distance connections.
In the brain, we've got like
I see.
like a small world network type stuff.
Yeah.
Can you deal with that by going into higher dimensions?
Yeah, let me see. So here basically, I uniformly randomly sample from the unit square, let's say, and then there's a parameter which basically says what's the size of my connectivity neighborhood. And in theory, for like one extreme is 0, and so no one's connected, and then you've got the other extreme where you're completely connected.
I see.
The big sort of thing to note is that in this derivation, everything is to order local edge length squared. Yep. So the limit doesn't apply if edge lengths aren't shrinking in some uniform sense as you take the continuum limit. But the interplay between the neighborhood size and the overall size of the embedding space is a very interesting area. And I don't have a complete answer for you, basically.
Cool. Thank you.
No worries.
Thank you very much for the interesting talk. I was wondering how you precisely triangulated the sphere and whether that is disordered. And is there a way to more orderly to triangulate?
Right. Yeah. So the Thompson problem is the problem of if you have n points on the sphere, what's the sort of which configuration minimizes the electrostatic potential of each electron, let's call it. And this is one way of producing the most uniform distribution of points. So there are exact solutions for a range of numbers of points, but yeah, there are different ways of tweaking it. Here, I've just uniformly, randomly sampled the sphere and then applied like a tetrahedralization essentially in 3 dimensions and then reduced it to the sphere. But there are many ways of doing this, and yeah, happy to chat later.
Hi, over here.
Hi.
Very good talk, thank you. In my corner of the world, we identify people like with the nodes more than with the continuum limit. And so, at least for us, or at least for ecology, it's important to be able to downcast these kind of continuum dynamics down to like a local neighborhood, because every person's situation is different, or every region. Is that the case in here? Is there a correspondence to get you from one to another and not sacrifice too much accuracy?
Well, it's all essentially about this. I mean, for fewer and fewer components, so people in your case maybe, this limit is going to be less and less representative depending on how large scale the dynamics are that you're trying to produce, trying to represent. But yeah, basically it's a matter of testing it in your individual case. The point is, if you have some system and you're wanting to look at large scale dynamics, a continuum limit could be useful. But if you want very local dynamics, it's going to be very difficult to have some coarse-grained description of that on the scale of the entire system. I remember your poster, so yeah, I'm happy to chat about it with you later.
Yeah, thank you.